So hey everybody. So I'm Levi, and I have been a long-term Crash member, not like the absolute original, but now like over a decade, which is kind of crazy to say. And so Crash Space, I guess the short answer is it's a clubhouse for nerds. It's artists, scientists, engineers. Like we've just been working on all sorts of projects. Like we just recently had an art show. We did a lot of stuff like mass producing PPE, for example, for COVID-19. We help crowdsource the fallout monitoring for Japan after Fukushima, and we also do a bunch of things like LEDs and things, and make art cars for Burning Man. So it's a whole wide gamut. And then I've been doing all sorts of stuff with that. I'm what was it? I joined Crash as a high school science teacher. Now I'm a postdoc over at UCLA, or air quotes at UCLA, because I've actually only been there twice since I got hired. And one of those times was just so my PI could just at least know that what I looked like in person. So anyway, I actually know Caitlin through, when I was still a graduate student, through ArtSci at UCLA. So I guess I'd like to just hand it over then to Caitlin and who she is and like what she's going to talk about with fungi.
Thank you, Levi. It's awesome to be here. I just want to say before I totally begin that I would love, I'm interested in interaction and like having a conversation more so than just fully talking at you for the next hour or so. So please interrupt me, ask questions. I'm really happy to just kind of, to talk. I don't think I can see the chat necessarily. So, and just interrupt me. All right. Oh, I keep doing this. So y'all see my screen again?
Yes.
Okay, cool. So just to, super briefly, my name is Caitlin Bryson. I work at the UCLA ArtSci Center, helping kind of manage and run the center and programming. I also teach art and ecology. I teach in the UCLA Sci-Art Summer Program. That's an amazing combination of all the things that I'm sure that you're interested in. But basically just really showing that art and science can be used as a complementary tool, as complementary tools for education, for learning, from students at a very young age into obviously well into graduate school even. So in graduate school, I studied art and ecology at the University of New Mexico. I received my MFA in 2018, and it was in graduate school where I really kind of was able to bring these two worlds together that I had been like really thinking were totally disparate things basically my entire life. So while I was at UNM, I studied in the Mycology department, and also in the fine art department, and worked with a lot of people, also kind of in other areas, just really kind of like facilitating connections between those two places. So that's a bit of my background. Now I'm a artist in LA, working on figuring out how to do that, especially thinking about how to do environmental public art projects. So that's something that I'm kind of like getting into now, but also just continuing to make things and grow mushrooms and do these performances that you'll kind of see throughout my talk. Cool. So I'm going to start with this video that was a collaboration between my partner and I that we made this last summer for a festival at UCLA as part of Ars Electronica. And I'm just going to read a little bit of an essay that I kind of wrote in tandem with this just to kick off. If it works, okay. Fungi are ubiquitous queer organisms in our environment that create and facilitate the world around and within. This unique queendom on the tree of life blurs the boundaries of worlds defined by humans. Often they exist and thrive within the fringes, between life and death, between singular and multiple, between sexes and identities, between the taxonomically distinguished, in harsh and impossible environments, and truly beyond our understanding of time. Fungi are permeable, promiscuous, and profound. Today I'm going to share with you my knowledge of fungi, which in comparison to their breath is microscopic. I will talk about what I have learned through working and collaborating with these organisms as an artist and activist, sharing what I believe to be vital information to us humans in the wake of our environmental, social, and political disruption. So I've worked and studied fungi for the last 8 years, gaining interest, really initially working as an organic farmer. And then I formally studied the sort of how to cultivate the biology of fungi while I was in graduate school. So initially, I was interested in um, how fungi could repair soil, and because working as an organic farmer, I was working in the desert, and so I learned about mycorrhizal networks and these things that seemed so fantastical to me at the time, and actually still do, but how nutrients were shared and distributed, how fungi were actually responsible for making fertility, or partly responsible, really kind of like the initiators of it. And I got really interested in how I could bring this sort of metaphor and also these physical happenings into my artwork. Um, while in graduate school, yeah, I kind of learned how to cultivate. I basically built like my whole studio is kind of like a bio art lab. I studied biohacking. I kind of created this whole environment for myself and for these organisms to really sort of like to just grow them and to watch them and just sort of see what they were telling me. And then I have gone on to teach workshops. I've taught workshops, yeah, all throughout the southwestern United States, and then also have kind of integrated that as well into my artwork. Really trying to think about how this knowledge and information can be shared and spread. These are just some of the things that I've grown and done, which a lot of the times, honestly, are just experiments. I From working and learning with fungi, I learned eventually that I was basically just setting up these environments for them, because I never really knew what was going to happen. And sometimes I would have these really beautiful moments happen, which here, this reishi mushroom sort of took over the aquarium that it was growing in and kind of ended up taking the shape of it. And it was very fascinating and beautiful. Other times I've had just like what mycologists and fungal farmers would say are like these terrible things happen, which is just like massive amounts of contamination, that I eventually found to be really beautiful. But I've sort of ranged the spectrum of having these like kind of really beautiful moments to these sort of like gross and actually um, uh, I guess very unexpected, like disgusting kind of things happen in my studio and in my work. And throughout this talk, I'm going to kind of go through projects. But first, we're going to start off with situating fungi. So fungal ecology, aka mycology, what are fungi? What is a mushroom? So I'm going to kind of discuss the branch on the tree of life, their morphology, their ecology, a little bit of phyla and classification, and then we're going to look at how fungi shape the world through nutrient distribution and ecosystems, network communications, how they create medicine, how they create fertility, and then into mycoremediation and waste reclamation. All of these this conversation is going to lead us into how we can work with these organisms for social and environmental justice. And that kind of leads us through the sort of like not only the metaphor and the sort of meaning of mycelium and of fungi, but also through potentially practical applications for healing. So what are fungi? Fungi are situated here in between plants and animals on the tree of life. They're distinguished. Um, we'll go here. Yeah, they're distinguished between these two kingdoms. Sort of, a lot of people think that fungi are plants, However, these distinctions really kind of separate them. So fungi have cell walls that are made of chitin and not cellulose. They're heterotrophic like us in that they acquire nutrients and food by absorbing dissolved molecules. They do not photosynthesize, but they very often work with and within plants, as you'll learn about, and really challenge these notions of genera, taxonomic classification, and this idea of individuality. So the first thing that we should talk about when we talk about fungi is mycelium. It's sort of its body. So mycelium is this white feathery substance that sort of grows in the substratum and within decaying material. That is the actual body of the fungus. And if we compare fungi to an apple tree, for instance, the mycelium is sort of all of its components. It's the root system, it's the trunk, it's the leaves, it's sort of the whole kind of system, the whole body of the plant. Whereas the mushroom, the things that I think most people often associate with fungi are actually just the fruit. So the fungi or the mushrooms are the apple. They are the sort of bearers of the genetic information, and that is how a percentage of fungi reproduce.
Quick question. So with I was wondering, I don't know if I'm remembering this correctly, but with mycelium, isn't there something really strange where you have multiple cell nuclei, but they're not separated into different cells or something?
Totally. Yes.
Oh, okay.
So this is another really, I mean, I could just make like a presentation on mycelium, but it's just so much. Maybe I can go to this slide. So actually part of the mycelium that you see in, in this image. So these you have these threads, these strands, right? And these are known as hypha. And in plural, they're called hyphae. And hyphae are a string of one cell tissues that grows sort of outwardly, right? So like if we think about how a carrot grows, cells are being stacked up vertically, whereas hyphae grow outward and they grow one cell thick. And what happens here is that there's this very like literally like fluid situation happening in the mycelial network, in the hyphal network, in that nuclei and the cell material sort of floats all around it. So you don't have these kind of like, just as you said, Levi, like they're not held within this sort of container. And so there's literally this exchange of this fluidity happening all around this decentralized network. So hypha grow at mycelium hypha, they grow at the leading edge. So right here you see this tip, and again, they extend outward. And then a mushroom is just this magical moment of mycelial condensation happening where all of the hyphae grow and condense into a reproductive structure, which is the mushroom. So only a small percentage of fungi, in specifically mostly the Basidiomycota family, reproduce and form what we call mushrooms.
You've mentioned percentages. About what percentage of fungi are reproducing using mushrooms as fruiting bodies, give or take? I don't mean to.
Juniper might know this answer better, but my understanding is it's only a small percentage. I mean, there's over 55,000 or 55. What is it? I don't know. I don't know the answer for that. It's a small percentage.
I don't know the percentage either, but it's like the teeniest, tiniest bit of fungi because most fungi are actually pathogens.
Mm-hmm. Mm-hmm. It's a very small percentage. And it's only really within, it's hard to say because also, like I said in the beginning, kind of like these distinctions that we have, especially within the taxonomic, like within the Linnaeus taxonomic system, like there's these sort of distinctions that make the classifications. But as mycologists and scientists are learning more frequently that I can't say that only the Basidiomycota, which is one of the phyla we're going to talk about, that those are the only that produce mushrooms, because now there are scientists that are finding that so much, that certain ascomycetes will form mushrooms that some kind of switch back and forth. So they'll be, I'll talk about this in a little bit, but they'll start off as a mycorrhizal soil-dwelling fungus, and then it will switch into a different type, and then it will produce a mushroom. So there's all kinds of different, there's a lot of different things happening that I think are really, it's a mystery. So I don't have an answer for you. I'm sorry.
No problem. Thank you very much.
Yeah. So fungal ecology, so back to kind of like how we classify things, or how we try to classify things. Part of that is, so what are they eating? Where are they living? And another interesting thing about fungi is that simultaneously their food source is also their habitat. So we have the decomposers, those who live in rotting material and who do mostly fruit and form mushrooms. We have symbionts, so those who live within other organisms and produce. They generally don't produce mushrooms, but some of them do. Also, of course, in that category, there are lichens, who are fantastic creatures with at least one alga or cyanobacteria and multiple fungi species living within the actual structure. And so these are, here we go, 5.1 million species. These are the three, the Glomeromycota, Ascomycota, and the Basidiomycota are the three that I have like still even, that's what I've studied mostly, and I have like a very small percentage of like knowledge about these three classifications, but I've studied them by not only like reading about and learning about them from mycologists, but also through working with them in my artwork. So part of like, that's how I have formed a relationship with these three. And this is kind of what I'm going to focus on today, because again, it's really kind of what my specialty is in. So yes, we're going to go through these three phyla to learn about how fungi and mushrooms are organisms that shape and have a significant effect on the world. So the Glomeromycota, mycorrhizal fungi, mostly. So these fungi form mutualistic relationships with plants, and there's a few different ways that they do this. Some of the sort of easiest, I guess, like the best way to talk about them are Endomycorrhizal fungi, who form relationships between plants via their root systems. So they tap into the root system. They have this mutualistic relationship where the fungi are extending the root network out like by tenfold, harvesting minerals and water from far off places, and immobilizing those nutrients for the plants in exchange for excess carbon that the plant is deriving from photosynthesis. So trees produce a ton, we'll just kind of focus on trees for right now. They produce a ton of sugars via photosynthesis. That is a lot. They have a lot of it. And so what they do is they store that excess carbon or sugars in mycorrhizal networks. And there's a lot of kind of, again, it's an increasingly advancing field of mycology, where it's just, I feel like every time I read a new paper, there's like something totally like amazing and fascinating happening with our advance in knowledge of this, and how ubiquitous this actually is. Over 98% of the Earth's plant life have these mutualistic relationships with fungi that, as Juniper was saying, they can be mutualistic and they can also be pathogenic. But we do know that there are, I don't know the amount again, but like hundreds of species of fungi at least, like living within a single plant. And we're also, we're ingesting that. We're eating, whenever you eat lettuce, or any sort of green, you're also ingesting those fungi. Suzanne Simard, I have a video here, but I think I'll just post stuff if people are interested. I can share video links. She's actually been an amazing scientist and scholar, really advancing this field, and also like delivering these really amazing talks that are like really helpful and easy to understand exactly like how this is going on. But it's been discovered, especially, that there's kind of this there's definitely like a communication happening between the plant life and the mycorrhizal networks, or the fungi living within them, in that there are these kind of distinguishing moments. There are these there's like determinations made when to deliver the nutrients and the carbon and when not to. For instance, you just kind of basing things off of just really kind of basic deciduous and evergreen trees. When deciduous trees lose their leaves, the evergreen species are kind of fostering more nutrients and sugars to the deciduous trees. And then in the spring, when those are starting to leaf out and they're kind of blocking them, then it's vice versa. So there's this kind of switch happening. Another sort of distinguishing factor within that is that it's been determined that that sort of the mother tree or the parent tree will send nutrients first to its kin and then out throughout the network.
Is there? I've heard about this with forests, like how there's because there's plant to plant transport of nutrients.
Mm-hmm.
But I've also heard that there's something really weird where if you, at least maybe for some species of tree, when you chop it down, but there's just the stump and root there, that stump and root are still actively involved in the transport network. Is that just because it's being mediated through the hyphae through the tree, or is there something else going on there?
Yeah, I mean, personally, what I think from what and anyone again can jump in here, but that is still, I mean, there's a ton of nutrients still left over in that stump. And so it's still very much an active participant. Like, again, it kind of like really distorts this idea of like living and dying in a way. But as it's, still there, there's still a bunch of, I would say, like nutrient information that is being kind of, again, like spread out through the ecosystem. And so very much still like an active participant within that. Again, maybe Juniper has something to add, but.
My 4-year-old has lots of ideas about this, but it's the roots. There's like the source and sink dynamics that are happening in the roots and the uptake. So sometimes it can just be a matter of just what's stored in the roots, and the mycorrhizae are kind of passive participants in dropping it between the different sources and sinks. But yeah, that stuff's super fascinating too. There's a lab at Berkeley where they look at that a lot. And they do it with redwoods. Todd Dawson's lab, looking at just the root mechanics of that.
Thank you. So the piece that I opened up with is actually, I was starting to, I've been thinking a lot about this, like this communication that's happening, essentially because like I'm interested in this sort, again, like this, these distinguishing moments, like how information is passed and how these like determinisms are made. And so, the way I've been an artist, I was classically trained, a painter, like the way that I think about things, the way that I understand that material is through making them. And so I'm also just like thinking about how we can begin to kind of, we as in humans start to literally like see fungi. How do we see this? Can we see this happening? Can we like have an emotional relationship with that, this sort of microscopic world? And so the piece that I made with my partner called Trans Relations was just like really, I feel like it was kind of a prototype for this deeper project that hopefully I'll get to talk about at the end, where we're looking at blowing this up literally, and thinking about how these communications are made. Can we translate those communications? Is there a way to like literally make them audible and perceive what they're what they're doing, what they're speaking about is a way to literally like tap into that system. So anyway, that's just what we were thinking about for this piece. But going on into the Ascomycota, common molds and endophytes. So even backing back up to what we were talking about, the mycorrhizal endophytes are actually a type of fungi that live within plant tissues. They live within the whole vascular system of the plant, but you can cultivate them with plant leaves. But these are also kind of common things that we like see all the time. The moldy, the mold that's growing on bread or on your citrus, Penicillium, Aspergillus, Trichoderma, things that like live in compost. They're all common molds, and they mostly don't produce mushrooms. They produce these things called asca, which are these like little cups, and that's kind of their reproductive structure. But they also produce tons and tons and tons of spores. And what you're seeing on your bread when you can like see the sort of green mold or the black mold or whatever, those are actually fungal spores that you're seeing. Yeast, of course, our friends, the yeast, who have had such a deep relationship with us throughout time, are also part of the Ascomycota. And Ascomycota are really kind of important medicine makers. So the most notable, at least in Western culture, being the discovery of penicillin by the accidental discovery of penicillin by Alexander Fleming, where the story goes that he left this petri dish. He was trying to find a remedy for strep and left this petri dish of the bacteria culture out in his lab and went on a 2-week hiatus. And here's the bacteria, and just go up. There was this developing mold growing on the petri dish when he got back, and he observed that there was like this kind of like this magic area right here where there seemed to sort of be a standoff. And then eventually the mold consumed all of the bacteria, meaning that what happened here was that the mold produced the mold and every mycelium, when they come in contact with a new food source, they exude all these enzymes. They secrete all these enzymes because they extracellularly digest their food through their mycelial networks. And the way that they do that is by basically just like they secrete all these chemicals that figure out like how do I digest this thing, this substance. In that process, it's like a suite of metabolites. So they're kind of a bad example, but it's like they're like exuding neosporin or something because they're trying to kind of like fend off what they can't consume right away. But in that process, they produce this medicine, and that's what antibiotics are. And just to pay homage to indigenous peoples all over the world who knew this way before Alexander Fleming. In traditional healing practices, it was common to put moldy food on an open wound, like a moldy piece of bread or something, because that actually would heal and subside the wound. So as I mentioned also in the beginning, like I had all these kind of experiments going on in my lab. I still do, that were really I was intentionally trying to sort of cultivate these, in the beginning of it all, I was like obsessed with mushrooms and wanted to like create these like gorgeous growing sculptures of like really sexy, seductive mushrooms. And like, I was very obsessed with kind of like this idea of beauty, this Kantian idea of beauty. And so much of what I did got contaminated. And at the time I was really like, I was working on ideas of contamination in the environment and like how we could clean up pollution and contamination with fungi. And then, back in my studio, like my things were getting totally contaminated themselves. And this became a really important teacher and like a parable for me. I think moving forward, like this very humbling parable about my role as a creator and what I, this idea of control and like what I have the right to do, or what like what am I doing as a human, especially when I'm creating these like super ripe environments for any microorganism to want to come and set up house. And so here's a piece. Eventually, I just kind of like displayed these works because I also was so fascinated with what was happening with them. After I did all this like intense sterilization and like tried to make these kind of closed off homes, that there was still something that would get in. And so there kind of became a whole bunch of work that was about contamination in and of itself. And I actually learned a lot about Ascomycota through doing this, and had these very, like these pieces, and eventually like really, really smelled though. But they were also really rich in nutrients, and I buried them all.
What is it? I mean, if you don't mind me asking, what does it smell like when it's digesting all of that?
I mean, it just depends. Like it was, I also had like, it depends on what the substrate was, honestly. Like, because I had this whole compost sculpture in my studio that was really just like pure, like rotting, like food, and that obviously really smelled like very eutrophic, like very like sulfury, gassy. I had like a bunch of explosions happen in my studio because like when I was first starting out, like my first year of grad school, I didn't really understand like gaseous exchanges and respiration. And so I had like made these like beautiful little glass sculptures, you know, and like put a bunch of compost and stuff in them, and they all blew up. And it was awesome, actually. Again, it was like right before like this always happened right before I had like a review or my committee was coming in or something. I'd have these like, you know, these contaminations happen. But it was also really awesome because it was just like, again, that like I'm working with living material. I'm obsessing about living material, and like I'm making these grand statements about like the world, you know, and like here I am being just totally like schooled by these little microscopic organisms that are really in control of the whole situation, you know. So like for that review in particular, there was like what I talked about with my committee was the fact that like the pieces had exploded, and it was like they were doing this performance themselves, you know, like there was like glass on the wall, there was like splattered like tomatoes and compost on the wall, and a bunch of glass all over the place, and you know, but there was respiration happening.
I have a question. The material descriptions is naturally dyed fiber with rabbit brush and indigo.
Uh-huh.
What does that mean? What am I actually looking at here? I'm not familiar with what rabbit brush is.
Mm-hmm.
Is this some kind of fiber that you're growing fungus on, or?
No, rabbit brush is this. So I should also say it's just there's so much to pack. I was telling Levi this in the beginning. Like there's so much to talk about. It's hard to kind of do it all. But whenever I make work, one of my main, like the main impetus is to make something that is completely biodegradable. This was part of like my process as an artist, kind of being trained as an oil painter and working with really toxic materials, like literally my whole life. Like I made this big switch before grad school actually, and kind of deciding that I wanted to make sure that everything I did like could somehow be put back into the ground and also be like an added nutrient into the ground. So I started working with fiber, and of course, like I'm obsessed with color because I'm an artist. So I got really interested in natural dyeing. Rabbit brush, you'll see throughout the work that basically everything that you see is like naturally dyed. Rabbit brush is this giant bush that grows in the southwestern United States. It's also called chamisa. But it's a flower. And then indigo, obviously, I'm sure you know what indigo is. So I've dyed fibers with mushrooms. I haven't grown, well, no, I've done a lot of fermentation too. But I haven't like specifically grown my own mushrooms to dye the materials with.
Thank you.
Yeah. Okay. So then moving into the Basidiomycota, and these are who I was intentionally trying to work with in this whole sort of process. They're saprotropics. So they live off of decaying material. They live off of and within decaying material. They decompose wood and other things, and they most often form mushrooms. one of the most kind of fascinating and like this very important thing that they do is they decompose lignin, which is one of the world's toughest substances. So it's the chemical that constitutes wood, and it's basically these very, very complex, tightly bound sort of carbonic chains that are super, super dense. And fungi have learned over eons how to decompose a substance in this very kind of unique way, where it's like not like they're going in and just starting to like, again, they're exuding all those chemicals and decomposing it. But instead, what they're doing is they're breaking into the wood and they're actually causing this oxidative process to happen. So it's kind of like this backdoor entry into like literally like going into these chains and oxidizing them and bursting them open, creating air pockets. And then in that process, a whole suite of other organisms come in and they start to, they're like, sweet, bunch of food, a habitat, usually a sort of a humid environment. It's like the exact situation that most microorganisms want to be in. And then the fungi can also, they eat those things. And so anyway, it's literally this whole like collaborative moment. But it's a very, very difficult substance to break down. And part of why this is so interesting is because there's so many other things that are toxic in our human-made environment that mimic this carbonic chain. So polymers, polycyclic aromatic hydrocarbons, crude oil, plastics, they all kind of mimic this same composition. And because of that, fungi are able to...
Quick, have you heard about this, I think, if I remember correctly, in the geologic areas of the Devonian. So there's a time when like trees, so like when lignin and cellulose arose, but before fungi could break it down. So there's like this era of history where there's basically just tons and tons and tons of preserved wood because like nothing actually could consume it yet.
Totally.
Have you? I don't know if you've read ever read any of that stuff or.
Yes, I have a little bit, and also, I mean, that of course that affected the entire atmosphere, right?
Mm-hmm.
There's all this stuff stacking up, and there's no again, like it was almost like these environments were of like my little compost sculptures that are just like super. There's no respiration. There's no activity able to transpire, like gaseous activity able to transpire. So it's like making the atmosphere actually super inhospitable. And yes, and there's all these facts too that like if fungi didn't go in and like do this initial work, we would basically have the entire earth would just be stacked of all of the material that's around us. There would be, especially in forests, and completely, it would not be a hospitable place for anyone. Just to kind of back up off of that, one of the things that I just read in regards to mycorrhizal fungi is that it's being studied and increasingly sort of proven that fungi were the first plant roots, and that those mutualisms that happen, between lichen and algae, they're actually kind of like the precursor to how plants were able to move onto land and begin the process of decomposition, etc. And that's also why it's now believed that the mycorrhizal or that endophytic relationships are ubiquitous in plants, and then that's actually like how plants were able to obtain nutrients and move on to the environment, and then eventually that roots begin to form around the mycorrhizae. And so this is a theory, but again, like the last few papers that I've read are all kind of like pointing to this. So that's like again, like another part of the talk that like we could talk about forever, is just like how fungi have helped the evolution of plants and animals.
I was going to say, I'm a believer in that, Caitlin, that plants make roots for fungi, but that the original fungal relationship of how it happened was that they were actually invading the plants as a pathogen, and they got leaky, and then that leakiness actually benefited the plant, and then you get evolution, and then you get the creation of these mycorrhizal symbioses.
We didn't eat no stuff.
Okay.
Cool. But yeah, So I'm excited about this conversation. I'm happy y'all are interrupting and talking, and thank you. So yeah, back to mycoremediation and just this potential of like oxidation and of these carbonic chains. So something that I was like fully focused on in grad school was remediation, which is the act of remedying something, you know, reversing it, stopping, especially environmental damage. There's bioremediation, which is using biological materials, and then mycoremediation, which is specifically using fungi to do this. Conventional remediation is like just really backwards in a lot of ways. Some conventional remediation strategies would be to, if there was a Superfund site, a toxic spill, or maybe some leaked, I mean, there's so many examples of this, maybe too many to get into, but you know, some nuclear waste, and to come over and to, you know, to bury it, put it in the ground, or to pour concrete over it, or to come in and physically remove those materials and send them off to somewhere else, which is obviously not dealing with a problem. There's tons of gaps and holes in all of those solutions. And so to, you know, I was a permaculture and biodynamic farmer. Again, like I'm very interested in systems and how things are cycling through, and especially how you can kind of create these dynamic systems by, again, just like putting the right ingredients in place and just helping facilitate a healthy ecosystem, right? So mycoremediation started to just like make a lot of sense. And this is really kind of the area that I studied for my graduate thesis work, and what I'm still kind of doing now, but now it's kind of tending towards like different directions. But there's a few different types of mycoremediation. So there's actually digestion, which happens with those things that are more similar to the lignin sort of chain. So with crude oil in particular, this is an experiment done by Paul Stamets in the 90s, where he trained a species of mushrooms, Pleurotus osteratus, oyster mushrooms, to digest crude oil. And they did. And eventually he has this experiment where he covered this oil spill with, you know, with substrate, with mushroom food, he inoculated that with spores. The fungi consumed the oil, they digested it, they grew. Birds came and ate the mushrooms, and a bunch of other insects came, and then birds came, you know, then the whole sort of system happens. And eventually there's all these images of this oil spill being completely like a little like grassy knoll. There's complications to all of these things, which we can talk about. But this is possible, and it is possible to do it. You do have to train the species. It's not, or you have to find a species that's very specific to that. But it's definitely possible. And then there's translocation, which is, unfortunately, there's not a graphic for this. That's a mushroom and just used to plant. But essentially, the mycelium will interact in an environment, and they will, depending on the material, they will pull that up or bind to it within their mycelial mass and locate it to their fruiting bodies, to the mushrooms. So in this case, you know, mushrooms are essentially hazardous materials depending on the substance. This most often works in heavy metals and installations. Sorry, I'm hearing sound. Does someone want to say?
Oh yeah, is it so? So like they're dealing with heavy metals? Are they chelating them when they?
Yeah, exactly. Yeah, I tend to not use, like, I've given this talk a few times, and I tend to not use that word just because most people don't understand. But that's exactly what it is.
Oh, okay.
Yeah, and so this works really well in areas where there's like water runoff. The installations sort of, this is an example of an installation which I've used, you'll see in a minute, where you just kind of make a big mushroom wall, basically, where water runs down, or the material would run down, and the mushroom would catch the fungi, and the mycelium. The basidiomycetes are like the ones usually used for this. So anyway, they'll catch that runoff and translocate it. But then again, the question is like, what do we do with all these hazardous mushrooms or mycelial mass? So that has to be dealt with as hazardous materials. And then lastly, bioassorption is one of the most effective means for mycoremediation, actually. And this works in aqueous solutions. And it's where the mycelial mass, the same thing, they will chylate the material into their tissue, into the fungal tissue. And you can, it works most readily with heavy metals, but you can literally just like, you can drag the mycelial mass like through a solution that has the material, and it will actually bind to the tissue. And this is a cool way of mycoremediation because you can use living and dead mycelium. In the last two experiments, it's very important to have this hospitable environment for the mycelium to grow. So like I was living in New Mexico, trying to do these experiments and, you know, have to go to great lengths to make these little biostructures to kind of house them. And that was really expensive and really difficult. So it's definitely better to be, you know, again, that's one of like the sort of the sub notes of all of this is that you have to kind of have hospitable environments to do this stuff.
If it works with dead hyphae, what exactly is it about? Is it just is there just something about the structure then? What is it exactly like?
I don't know if I can answer that fully. I was doing an experiment, was working on this project for, I'll talk about it in a minute, but for Los Alamos National Labs with mycologist Peter McCoy. And we were looking at hexavalent chromium remediation through essentially making these little beads of dead mycelium that the hex chromium would bind, it would just bind to it. And again, it was just like, it could be suspended in the aqueous solution for like not even very long. And it was through this chylation that was happening. But I don't know exactly like the biochemistry. I can't speak to that fully right now. But
I could say something. I don't really know much about the biochemistry, but when I think of the hyphae, I just think of so much surface area, which is why they're so much more effective than roots at absorbing nutrients. So when you basically got like the teeniest, tiniest, finest net to comb. But so clearly, I don't remember what's happening with the biochemistry, but yeah, it's just like a big part of it is surface area too.
How am I on time? Because I'm at an hour right now, and
There's no. I mean, the only like we have another thing starting at 6:30, but that's about it.
Okay.
Yeah.
So I have like a couple things to read, just part of like this work, this whole project that I did. And I just want to read a little bit about it, but it all kind of like leads into this project that I did for my graduate degree. And really kind of thinking about this idea of where we are now and how, like, thinking of this time as compost in a way. I think of all time as compost, but really just kind of thinking that we're in this like this place of precarity where it's kind of ripe for decomposition and nutrients to grow into a new era. And I was really inspired by Anna Singh and Donna Haraway, and you know these sort of people who are kind of like also really talking about this stuff. Actually, I think I'm just gonna keep going, and if anyone wants to read my writing and stuff, I can put it on. It's on my website. But really thinking about how precarity is again like this reality that we're all kind of enfolded within. And that the decay of the systems of our world are actually, it's alarming and it's really uncomfortable, but it also is it's positioning us for fertility and for growth. You know, nothing better describes this situation than seeing fungi fruit off of rotting material, or seeing all these sort of magic moments happening even in a compost pile. And I think, again, like this is where this metaphor and the symbolism like really is driven home for me, like to see these things like literally happening sort of in front of our eyes, and like how can we use this not only as a metaphor, but again also as a means for kind of dealing with these situations we've put ourselves in as humans. So in the book, The Mushroom at the End of the World, Life in the Midst of Capitalist Ruins, Anna Singh talks about the matsutake mushroom, which is a prominent gourmet mushroom that sort of forms this really interesting relationship with pine trees. It's a mutualistic relationship, a mycorrhizal relationship. So matsutake is actually a mycorrhizal fungi that forms mushrooms. But the whole thing is very interesting, and this kind of, again, this like this example of how fungi form adaptations, because pines and matsutake thrive in disturbed ecosystems. They really want these barren soils, these really highly mineral soils. And this kind of situation has mostly happened because of deforestation. And she traces the life of this mushroom from its ecology to its economy as one of the most prominent sort of gourmet mushrooms in the world, and how it's kind of created this entire kind of like literally underground economy for people who are who mushroom hunt the matsutake, who sell them, and what is the market. And it's literally a whole network of this important system living in the wake of sort of huge disturbance. And it's not meant to sort of be exploited as this superhero kind of championing human greed, but really as this idea of how we can adapt to these situations that we've been in, that we've produced, and how we can sort of how we can form relationships, basically, and get ourselves out of this place, I guess. And I'm very interested in this. I'm very kind of interested in this idea of kind of like of radical hope, and finding hope in the dark. And Juno Diaz and Rebecca Solnit, two scholars who I'm very, very interested in their work, they both speak to these things. And for this project, I was very inspired by their writing. I'll read a couple quotes from both of them. So Diaz describes radical hope as not so much something you have, but something you practice. It demands flexibility, openness, and imaginative excellence. We need to bear witness to what we have lost: our safety, our sense of belonging, our visions of our country. We need to mourn all of these injuries fully, so that they do not drag us into despair, so that repair can be possible. And Rebecca Solnit proclaims, hope is an axe you break down doors with in an emergency, because hope should shove you out the door, because it will take everything you have to steer the future away from endless war, from annihilation of the earth's treasures, and the grinding down of the poor and the marginal. Hope calls for action. Action is impossible without hope. Every time I read that, I get emotional. But I like this, like, and I was making this work even just a couple years ago, and still, now it's just even more important. But like, what is this? How can we be hopeful in this world that we're in right now, and how can we actually activate that hope, and like, what does that mean? And I love this idea of being radical. Like, as a radical mycologist myself, it's like using the lessons that are all around us by these great microorganisms and these great teachers to kind of to not solve these problems, but to compost them. So this project called Gently Radical Changing was really about that. I worked with a really incredible group of women from an indigenous organization in New Mexico. They're called Tewa Women United. And I worked with the environmental justice sect of that for this whole project. It was this big collaborative project with them. And we were specifically working with and dealing with environmental injustice issues still stemming from the development of the atom bomb in Los Alamos, New Mexico. And so Espanola, New Mexico, which is where Tewa Women United is based out of, sits right at the base of the watersheds. The image is really light. I don't know if you can see it, but this is a topographical map of the region. And Los Alamos and Espanola are kind of two sister cities. And Los Alamos sits at the top of the watershed and Espanola is at the bottom. And basically, since the development of the atom bomb and the industrial war complex at Los Alamos National Laboratory, and then now just these unfolding sort of, projects and wastes that have still remained there, Espanola has really faced the brunt of all of the basically all the issues that have come out of this time. Espanola, there's some crazy statistics in that Espanola is like the third poorest county in the United States, whereas Los Alamos County is among the most affluent in the entire country. And Los Alamos acquired the area through eminent domain in the 1940s. So bought the land, built the labs, moved all of the people out, and they called this so-called, it was non-populated territory. So they said, in all of I looked at a bunch of historical documents from this time, and basically just said that there was no one living there, and barely, if they paid certain people, a lot of people, people of color were left unpaid to move out, and then sort of, everyone, then they set up the secret mission and everything happened. But Tewa Women United, the city of Espanola and the surrounding area is still very much dealing with all the fallout from that time. And of course, this is a type of environmental racism. This is a type of slow violence that is still affecting the bodies of the people, the water, the plants, the land. There's all kinds of things happening and a lot of waste still being totally mismanaged. So I'm not sure why these pictures are so pixelated, but here's just an aerial view, just so you can see a little bit of the geography of the region. Down here in this image, this is the labs, and this big chasm here is the Rio Grande. The lab was, initially draining most of the toxic material straight into the river. There's also all of these sites that are waste sites all throughout this mesa here. And this is all, of course, it was indigenous territory. When I worked with Tewa Women United, we did a lot of tours for the labs, and actually there were we passed by all these burial grounds and these kivas, which are these big sacred pits that actually, in the beginning of the project, that were being used as waste dump sites. So these like literal sacred urns and areas were being used to like pour the toxic waste into. And all of this has, throughout the last 60 years has come up, but there's still, again, like a ton of it that's still erupting and that is still being dealt with. So I had the pleasure to work with them really closely for 2 years on this project. And what we did is created 3 different sites were targeted, and I researched a specific strain of fungi, researched the toxin, and then made an artwork that was sort of focused on that specific contaminant. For the duration of my thesis show, the work lived in the gallery and fruited in the gallery. Again, these were made from natural materials. They were made from organic linen and plants that were dyed, that were harvested from the areas for the most part, and then stuffed with the mushroom and the substrate. And then, they lived in these little biosculptures. And then eventually they all were buried with a group of people in the communities, on all led by and like facilitated by Teo Women United and like my main collaborator and deep friend and mentor, Beata Sosi Pena. Part of that, we also did workshops where I was like teaching just about mushroom cultivation and how you can kind of like hack these things at your home. We did a really cool workshop and installation in Espanola, which is pictured here, where everyone made all these mycelial bricks and then got to take those bricks home and watch them grow, and some of them fruited, and then we all came back together and put them in the ground for this water runoff into a garden. But that was really kind of an active part of the work, was like teaching like how to do this at home and how and why you should do this, and also just the amazing world of watching mushrooms grow. But kind of the most important like facet of all this work, of this 2-year project, was of course the connections and the deepening relationship that I formed with Teo Women United. And eventually I was brought on their team as an activist and a member. I think my official title was consultant, but I don't really do much consulting. But it was to show up and testify in court in a public hearing against Los Alamos National Labs for specifically the hexavalent chromium plume, where there's this big hexavalent plume in an aquifer right below Espanola, and it's still being completely mismanaged. There's no responsibility taken. There's like all these conventional remediation solutions that are really kind of at odds with indigenous cosmology and how they want to treat and remediate their land. And so that's why bioremediation and mycorremediation are really actually in sync with the idea of, again, working collaboratively with the land and facilitating systems to actually facilitating ecosystems to create a sort of cyclical translocation of the material. And so I worked alongside them for 9 months, kind of preparing for this hearing, doing a bunch of workshops and talking to people in the community, from like tiny little kids to the community elders, so that everyone was kind of on board for like, what are mushrooms and how do mushrooms, like what is this and what is this world? And so we did this big kind of like education initiative, as well as just like literally showing up in court with them and being there the entire time and speaking out and testifying and proposing a pilot project. The crazy story happened is that this was all super corrupt. Maybe it's not that crazy, but eventually nothing came of this because there was corruption in the board, and basically the whole case was dismissed. This was a project that Teo Women United and this another organization in New Mexico called Community Coalition for Clean Water was working on for like 2.5 years, and eventually nothing happened. And the chromium plume is still just like it's expanding out into the groundwater in Espanola. These people have had staggering health impacts from everything that's gone on at Los Alamos National Labs. It's deeply kind of the complexities of this whole thing are so deeply embedded because a lot of people are employed by the labs. Within the communities, there's a lot of rift between families. It's basically just this seismic fission that has happened from all of this. And Los Alamos National Laboratory is not especially now, there's a lot of really interesting and very, very important research that's coming out of that place. And actually, in this process of working at UNM and working with these people, like I met a lot of friends, a lot of scientists there, but the blatant irresponsibility and like lack of accountability for these things that are still happening, and these health impacts that are still like greatly affecting this population is, it's awful. It's absolutely awful. And part of the work, the artwork, was really to just kind of bring this all to the forefront of, of my sort of art world, bringing the stuff into the gallery. And yeah, I was certainly coming at it from a very privileged position as a white person. And so it really kind of was important to just be like, I listened a lot. I tried to just really facilitate working, hearing the voices of the people who are actually facing the issues. And so for my exhibition, I don't have pictures of it, but for my exhibition, there was all these different events that happened, and many of the people from Te Ao Women United came and they actually spoke. There was like poetry readings or performances. And then, again, I was like actively kind of like in the community working with education about mycoremediation and just the potentials of working with these organisms for healing. So these are just kind of just images of various projects that I did with them and with others. And always kind of having these community efforts towards these installations and treating that as a performance, and specifically as like as a ritual where we're putting something in the ground and trying to transform what's happening.
Do you for each of these, do you have like a particular fungi in mind for the bioremediation that's like targeted, like, oh, this one's for hexavalent chromium, this one's for this bacteria?
Totally. Yeah. So that's part of it too. And I mean, it's all a process for me of, of learning these things. My intention and my dream is really to, I was talking to you about this, Levi, like I want to do a PhD and I want to study ecotoxicology, where I'm really kind of doing, also kind of sounds like what you were saying, Juniper, but I want to do this research more specifically in like a lab situation, but really be disseminating that research through art projects and through these community projects where, like, again, these issues are brought to the forefront of dialogue. But on my own, I was working with mycologists at UNM and also with Peter McCoy on kind of targeting specific, the contaminant and the specific species of fungi. Because again, that's like a whole nother world, is that like depending on the contaminant, it's like a specific type of remediation, it's a specific species of fungi. It's also not just like one blanket solution where it's like, oh, we're just going to put oyster mushrooms on this oil spill. It's like there's a whole bunch of other organisms that are important within that situation. It's not just the fungi. So really trying to, again, like create, it's really about, I think, like the habitat and the environment and setting up these situations where the players, the microorganism players involved can have the nutrients they need, have the environment that they need, and then potentially the remediation can unfold, but part of this all is just that for me too, as an artist, I don't have like qualitative data from any of this. It's a lot of quantitative data and these very meaningful things that happen to me, like the most meaningful things in my life, but I don't have like hard data about what, if we removed the RDX from the soil at the farmer's market. So very much in my practice right now, it's all still a metaphor. Yeah. Okay, now on to racial justice.
So but you're thinking about doing like a PhD to actually to be able to get that kind of data.
Yeah.
Okay.
Yeah, I think, that's kind of where I'm I'm in the in-between right now in so many ways. Like I want to deepen my knowledge. I also just, I really want to work with people. Like my art practice has like never really been a solo thing. And especially like, I want to work with a team of people. I want to work with scientists and labs and really be testing these things, but also be talking about them. Like this, this test site is also a public art site, for instance. And like, we can come and look at this, like we can see what's happening. It's not behind closed doors. It's also beautiful. Like because you're watching something decompose, maybe like we're able to watch this change happen, like even if it's a metaphorical change. Something I didn't mention, but like on all of the, I call these like mushroom pillows, but on all the mushroom pillows, like from the work with Beata and Tewa Women United, I embroidered this imagery, and every like little quilt was very specifically like had imagery that I worked with Beata on, like what is this, what's on, like what is literally like the image on this thing, what is the mushroom eating, and down to this spiritual aspect of that as well. And in my like Western trained like white brain, I was like, oh, we want to put the bad thing on there because then it's like the mushroom's eating the bad thing, right? And then we're like visualizing that going away. But Beata was just like, no, that I want to see, like we practice like their cosmology is like positive thinking, and I want to see what we want to manifest beyond that imagery, like that process of decomposition. Yes, like I want to see that on there, but also like we want to manifest, like we want to pray to the corn mother, we want to have our wheel on there. I want, the 4 directions need to be located on there at all times. Like we need to have this imagery because that's actually the food, like that's what this is metabolizing and eating and like transpiring into the environment and to the people. And so that has also played a huge role in all of this. And I think that if you scale that up to be like these public art installations, like then you have all these different things at play. You have the voice of the people, you have this transformation happening, that you also have this again, this like spiritual kind of like, I would say like spiritual decomposition, but decomposition in like this very beautiful way. When I think of decay, I think it's like the ultimate state of being, honestly. And so like, I think to have like this like spiritual decay happening where it's like it's being activated in the environment, it's being activated through the people and through the artwork, like that's actually like where transformation and where justice really comes from. This was an artwork that I made this summer, really kind of talking about these things. And I don't have, the space that I have right now is super, like, it's literally really small and limited. I don't have a studio. Like my studio practice is kind of non-existent right now. But it felt really important to, to obviously do something at this time. So I made this pillow embroidered words on it that were part of this like small manifesto that I made with my partner, and also all of the names of the people who have been killed by the police since 2014. Again, really thinking about these names like as a prayer, essentially, and that these people and these names and these words are a prayer towards letting this all decompose and be composted and be food for change and transformation to actually take place in our world today, both environmentally, politically, socially, all of those kind of working together. Last thing I want to say is just that another thing that I've talked to Levi about, and that back to this project of trans relations, is thinking about how we can also work with these systems to like literally listen to them and to try to understand what they're seeing and perceiving, or sensing and perceiving in the environment. And when there's a signal that comes in a mycorrhizal network, it's releasing these chemical signals, and a stimulus or an antagonist or whatever, like they're releasing these certain signals which can be used as electrochemical signals that we're sort of tapping into, or we're trying to understand. Like I want to also study this and think about ways in which that we can work with them, and like towards understanding more of what's happening in our environment, but also as kind of like a data sensory experiment. And this is very much, I've talked to Levi about this a couple of weeks ago, but it's like very much in the beginning stages, but another thing that I want to like potentially do a PhD for or start researching. So if anyone has thoughts on that or is interested in that, I would love to talk with you more.
This is this is the idea of like like what we're talking about with using electrodes to record the signals from the hyphae, and then we're talking about doing machine learning on that. Yeah, okay. Oh, Lindsay, you have an idea? Oh, okay. Never mind. So,
So that is my talk.
Lindsay was saying, wasn't there a guy who used mushrooms to play a synthesizer?
Yeah, there's a few people, and that's actually how I got into it. But then, like, even there's, yeah, so yes, it's really cool because you can hook it up to a MIDI and you can play the synth, and the sound actually, that's in the video is it was stressful. We couldn't hook it up, we couldn't do it, but we were modeling the sound in that piece translation off of those sounds. But and that's kind of my first entry point. I'm like, oh well, but how do you translate the sounds? Like, how do you distinguish sounds? So it seems like maybe something that's not, like not through a synth, but through what Levi was suggesting, through machine learning.
Well, you can encode sound in just a frequency and intensity map.
Mm-hmm.
You can simplify it that way, and then you read in, like, because each frequency, like, basically each pitch is going to have a certain like volume to it.
Mm-hmm.
What you can read in is like your set of inputs and say like, okay, here's, or like, or just here's like the output from the fungi. Like this is what I'm recording, and the
Mm-hmm.
input will be all the environmental data you're collecting at the same site.
Mm-hmm.
Saying like, okay, if the fungi is really a machine that's learning this, it's taking in environmental data and
Mm-hmm.
outputting these signals that we can encode as audio.
Mm-hmm.
And using something like random forest or whatever, like a machine learning model, you can say, okay, what system will actually take this environmental data and transform it into this like sonic landscape? And it's something that you can train it on. Because okay, he's got a thing called Data Room that he can talk to you about, but it's basically he's trying to make it easier to make code. So
Yeah.
but,
I'm taking that.
yeah.
I like, that's all super new. It's so new to me. It's like I don't even really have the tools yet to understand it, but that's
I think, I guess when we talked about this last time, I think that would be the way to do it.
Mhm.
Or at least like for my training, that's kind of like what comes to mind.
Mhm.
Of course, then the question is like, okay, like who we got to get funding or whatever, and I need
Yeah.
to hammer out, like, I guess where I'm at come September.
Mhm.
So that's what I'm still working
Yeah.
on.
No, totally. I mean, that's good. That's a realistic time frame. Also, just it's like more research and testing even too, and just kind of it feels like such a intangible project right now. So yeah.
But I think how you described it, it makes, like, it's concrete to me, like how you would, okay, go get the data, transcribe, like convert it, the soundscape data to
Mhm.
a pitch and volume map.
Mhm.
That is like what you would use to, train your machine learning model.
Mhm. Mhm.
So,
Yeah.
and there's different tool. The one I would come to mind would just be random forest, just because I've worked with it, but I know there are other ones.
Mhm. Mhm. I'm taking these notes too. Whew.
That was cool. Josh, are you there?